Hello, everybody, and welcome to the latest installment of the CDX Engage webinars hosted by Biodesics. This webinar is entitled A New Era in Precision Medicine, uncovering the depth and breadth of the plasma proteome with novel proteomic technologies. This is an online event designed to bring you with the opportunity to explore how new technologies are enabling researchers to see deeper and broader into the plasma proteome than ever before. To briefly introduce myself, I'm Noemi, a member of the CDX Engage team here at Hanson Wade, and I will be chairing today's session where we will be joined by two expert speakers from Biodesics and Seer. This webinar today will consist of two presentations, followed by a Q&A where you will be able to ask your pressing questions to the speakers, together with some quick interactive poll questions to let you provide your feedback. Today, we will hear first from Daniel Hornberg. Daniel is a principal scientist at Seer, focusing on the development of transformative solutions for biomedical multiomics at the intersection of bioinformatics, mass spectrometry, and nanotechnology. In 2009, he co-founded a company that developed large multi-enzymatic complexes leveraging extremophile bacteria to degrade resilient substrates. During his postdoc studies at Matthias Mann's lab at the Max Planck Institute of Biochemistry, he investigated proteome perturbations associated with neurodegenerative disorders such as ALS and Alzheimer's. Dr. Hornberg holds bachelor's and master's degrees in molecular biotechnology from the Technical University of Munich. But before handing over to Daniel to begin his presentation, I would like to first start with a poll to get everyone started and involved. You will have about 30 seconds to submit your answer, and then I'll show you the results so you can see how your response compares to the rest of the listeners joining us today. So, do you currently utilize proteomic technologies for clinical research? A, yes, B, no, or C, not currently, but interested. So you will have around 15 more seconds to vote. Perfect. So I'm going to close the poll now. So 56% said yes, 18% said no, and 26% said they're not currently using them, but they're interested. So moving on to the second poll. As before, you will have around 30 seconds to vote. Perfect. So I'm closing the poll now. And finally, the last poll. What areas of research most interest you for using proteomics? Disease biology, new drug target discovery, drug mechanism of action studies, biomarker of drug action, or predictive biomarkers for drug efficacy? So you'll have around 30 seconds to vote for this one. I'll give around 10 more seconds. Okay, great. I'm closing the poll now. And as you can see, most people voted for predictive biomarkers for drug efficacy. Great. So now, without further ado, I will hand over to Daniel Hornberg, Principal Scientist at Seer, to begin with the first presentation. So Daniel, please join me on stage now by sharing your audio and video.
Hi.
Hi, Daniel. Perfect. I can see your slides are up, so if you can go on full presentation mode for me.
Great.
Yep. There you go. I will see you after your presentation.
Thank you so much for this really kind introduction, and welcome everybody. Good morning, good day, good evening. I'm really glad that we can now today talk about Proteograph and new proteomics applications to dive really deep and rapid in a scalable manner into the plasma proteome. And throughout the next 20 or so minutes, I would like to introduce to you our technology that we develop here at Seer that enables people to get really new insights in that really important and very complex biospecimen. But before we get started talking about the technology itself, I'd like to take a few minutes to just give you a brief reminder of the utility and complexity of the proteome, in particular when we compare it to the genome and transcriptome. So as we all know, we have around about 20,000 genes and already quite a few more transcripts that derive from these genes, including all the splicing events. But when we look at the proteins, they are really far more complex. So we have, of course, different isoforms that are derived from different RNA processing processes. And then on top of that, we add the post-translational modifications like phosphorylation, glycosylations that can switch on and off proteins and dramatically increase further the complexity of the proteins and the proteome itself. And we would call these entities, these molecular entities, proteoforms. So that's a functional entity that can be present in form of a proteoform in the form of a protein with a certain post-translational modification, or as a certain isoform or a combination of both. And on top of that, we have, of course, the entire interactome of proteins. That means which protein interacts with what other proteins in what context. So the spatiotemporal information of the proteome is, again, yet another important addition to the complexity and also to the defining essentially the functional properties of the proteome. Now, given all that complexity, and in particular when we're looking into plasma, that comes with a huge challenge, because it's like, first of all, a very diverse biochemical space, right? It's not only for nucleic acids, but it's 20 amino acids that then can assume a certain three-dimensional fold and can have modifications. So there's a really huge diversity of biochemical properties that makes any assay actually more challenging. In particular, when we look at the plasma proteome, which conceptually is very interesting because blood in general connects to literally or almost all tissues and may contain signatures for all types of diseases and physiological states. But the challenge here is that the dynamic range is extremely large. So we talk about more than 10 orders of magnitude, whereas the few high abundant proteins make up a majority of the mass in your sample. In fact, 22 proteins make up 99% of the mass in plasma. That means that 99% of the signal you see on a detector is actually contributed by only 22 different proteins. And while we might find some interesting signatures also for these high abundant proteins, we are often interested in what happens down the dynamic range here, down to the interleukins, for example, and the low abundant proteins that may indicate some immune cell signaling, that may indicate some tissue damage. And that is really hard to detect with any downstream detector. Now, there are ways to dive deep into the plasma proteome, unfortunately not at scale, that have been used in the past. Usually those approaches include multiple steps, for example, abundant protein depletion. So you use antibodies against high abundant proteins, you deplete them from your sample to essentially not oversaturate the downstream detector, such as the mass spec. The problem with that is, first of all, it's like a pretty complex step, in particular when you are interested in different species, because you need to have the antibodies against different proteins that are high abundant in that particular sample. And then also you remove potentially some proteins that are carrier proteins for other lower abundant proteins. And that means that you might lose some interesting signal by depleting the high abundant proteins in your sample. Then those laboratory workflows include often peptide fractionation. That means that you separate the peptides before you inject them into the mass spec by high pH fractionation, for example, to decomplex individual injections and essentially make the work less, make it easier for the mass spec downstream to map out what is in your sample. Now, unfortunately, that not only comes with a lot of different steps that make scaling almost impossible, but it also means that there's a lot of variation introduced in every single step, as well as the problem of transferring the results from one to the other lab, because as you can see here on the right-hand side, It's a pretty complex equipment that you can set up, and there are different flavors how to set it up. So it makes comparisons, especially for clinical proteomics, very tough, and sacrifices either like the throughput if you don't do these, if you do these steps, or the depth of insights if you would just go with a neat plasma. So basically just looking at the top few hundred proteins. At Seer, we address that challenge in scaling and accessing the information of the plasma proteome by using a novel technology that, in an oversimplified way, essentially condenses the complexity of a lab down to a nanoparticle. Now, how does it actually work? So the core of our technology are these nanoparticles that, in the simplest design concept, are a superparamagnetic core, or composed of a superparamagnetic core. That means we can easily handle those particles in an automated fashion, and then they are surrounded by a glass shell. And this glass shell can be modified virtually in unlimited ways by attaching small molecules, larger molecules, even up to proteins. And the key here is that these surface modifications will tune the physicochemical properties of these particles in a highly engineerable and defined way. Now, when we introduce these particles in a complex biosample, for example, blood plasma, what happens is that a so-called protein corona forms. And this protein corona forms based on the physicochemical compatibility of the surface properties of the particles and the surface appearance of the proteins. And in fact, that happens at the proteoform level, because we can interrogate here native proteins, and depending on how they look to a particle, they bind. Now, in contrast to targeted approaches where you would have to define a certain antibody against a certain epitope to catch and capture particular proteins, these particles are, although very specifically interrogating certain properties, they generically bind across thousands of proteins. So you don't have to predefine a particular particle just targeting one protein, but with one nanoparticle that's functionalized, you catch hundreds, if not thousands, of proteins. But how does it help now to dive deep into the plasma proteome? So now imagine you introduce these particles into a plasma. What now happens, as I described previously, is that you form this so-called protein corona. And this protein corona is formed in a slightly oversimplified explanation based on two facts. First of all, the affinity of a protein to that surface, and second of all, the concentration of that protein in your sample. So it's essentially affinity times concentration that determines the composition of that corona. So concentration will always be a signal that you capture with the antibody. But since you have also leveraged the affinity, that means that you have one property that is independent of the concentration that essentially renders low abundant proteins more visible to downstream detectors, because the particle can interrogate throughout the entire dynamic range. Now you can assemble multiple particles into a panel so that they synergistically interrogate the entire proteome down to the lowest abundance range. Now, at Seer, we have integrated that system into the so-called Proteograph Suite, which is consisting of consumables. That means all the nanoparticles you need for a deep dive into the plasma proteome, together with all the enzymes and buffers you need to get you from your sample, from your biosample, to your peptides, in conjunction with the SP100, which is a liquid handling automated platform that takes care of the entire sample preparation procedure in a highly reproducible manner. So you essentially just load the deck in around about 30 minutes, and you end up with your peptides that you can introduce into your mass spec after 6.5 hours. Now, we are agnostic when it comes to the mass spec, so we can work with Bruker systems, thermo systems, SciEx systems, whatever you have already set up in your lab. And the great thing about that is essentially that if the vendor comes up with a new mass spec, our technology scales synergistically with the increased performance of the mass spec detector. And it helps essentially every mass spec detector, every generation, in having an easier job to dive deep into the into a complex biosample like plasma. At the end, we also have a data analysis platform that allows you to get from the raw data to the peptides and protein quantifications. Under the hood, we are using state-of-the-art software solutions like MaxQuant. And on top of that, it provides you with a very detailed QC that is part of our kit. that allows you to compare performance of your mass spec workflow from now to next year, as well as across labs. So to really enable people to do consistent, deep proteomics at scale and have something that can really be translated from lab to lab and also over time. Now let's have a look at some data. Here we compared the Proteograph assay that we conducted on poor plasma with 5 nanoparticles and compared the very same sample interrogated through a deep fractionation approach, depletion approach, and the neat plasma approach. So neat plasma is essentially just like taking the plasma, doing a very simple digest, and see what we get. All of that was then subjected to the same mass spec workflow with relatively short gradients, so 30 minutes DIA. That's around about a 20-minute elution of peptides. And when we look at the number of protein IDs we get at 1% FTR here, we see that we get around 1,600 proteins for Seer Proteograph. That compares to around about 700 proteins with a deep fractionation approach and about 300 proteins when we just do a neat plasma and digestion and injecting those peptides. Now, asking the question where we gain the additional proteins when we look across the dynamic range, looking from the high abundant proteins up here to the very low abundant proteins down here, using a reference plasma database. What we see here is that we dive much deeper into the plasma proteome. You effectively see here that we sample even down to the lowest abundant proteins. We identify proteins and quantify those. And you have to see that in contrast to the neat plasma, we really can see that you start missing a lot of the proteins that are supposed to be very low abundant. Now, of course, if you can do proteomics at scale as well as at depth, it doesn't necessarily help you for biomarker discovery if your precision is basically all over the place. So when we look at the peptide CV, which is our measure of precision here, we see that with a Seer platform, we get a pretty good precision of around 17% on average, which is much better, in fact, than the deep fractionation and the depletion approach. But of course, you have to keep here in mind that with the Seer platform, we look across 1,700, close to 1,700 different proteins. So at very low abundant signal, actually. Whereas neat plasma, for example, it only takes or accounts for the signal, or the CV only represents a signal for the very high abundant proteins. So this is the much harder case for Seer Proteograph to get very low CVs. And given that, we do a pretty good job while we dive deeper into the proteome, we also maintain a high reproducibility of quantification. Now, I'd like to give you a little bit of an insight into how we can use Seer Proteograph to identify new putative biomarkers in a clinical pilot study. So last year we published a paper where we investigated 141 subjects or a cohort of 141 subjects that were composed of early NSCLC cases and healthy controls. And this whole study was conducted within 2.5 weeks, starting from the sample preparation to getting the mass spec data ready for analysis. So throughout the entire study, we quantified around about 2,500 proteins, which compare with Seer Proteograph, which compares to around about 400 proteins using a conventional depletion approach. Now, this study was published last year in Nature Communications, and one of the things that we tried in that study successfully is to build a classifier based on the signals and the quantifications of the proteins. And you see here the performance of the classifier looks actually pretty decent with an AUC of 0.91, distinguishing between healthy controls and early NSCLCs. Of course, that study we would consider still a pilot study, 141 subjects, although for deep proteomics, probably unmatched nowadays. It is something that we actually think should be done at thousands and tens of thousands of subjects. And Seer Proteograph is a way to actually enable that. But nevertheless, we can think about, is there a there? Do we find some indications of putative new biomarkers? So we looked at the top features that contributed to the classification of healthy controls and NSCLC subjects. And what we see here is the desired pattern that we always like to see in omics, where we have a mix out of already known candidates putative biomarkers indicated here in red based on the open target score, and some of the proteins indicated here in gray that have never been implicated in the disease. So is there in this pilot study? I would say yes, but of course we aim for much higher studies, much larger studies with our pipeline. Now, the beauty of doing unbiased, untargeted proteomics, which means using mass spec as a readout technology, is that you're not only getting proteins, but you're essentially getting a much more granular view on what happens in your sample. That means you're seeing different peptides that map to different proteins that actually can be indicative of certain isoforms or the presence of certain post-translational modifications. So as an exemplification, again, we looked at that data that I just presented to you, and now we basically look at the peptide level. So on average, we get around about 8 peptides per proteins that we identified in that study. And of course, for some of the larger proteins, we get way more peptides. But that allows us actually to now take a next step and see what is really happening at the protein isoform level, for example. So one of the things that we can look at is like taking one protein here, finding the cases where for a particular protein, we see differential expression and abundance of certain peptides that may speak to the presence and absence or regulation of particular isoforms. So when we do that analysis comparing NSCLC and healthy controls, we come up with a list of proteins that we see here on the left-hand side that have differential expression of some of the underlying peptides. And again, like we have a nice mix of proteins that have been implicated in the disease already indicated by the open target score, and those that have not been identified to play a role in cancer. Now we can map those proteins down to the dynamic range here again, and there are two things that we can see. First of all, we get a couple of proteins that have some interesting patterns when we look at the peptides, but also we see that some of them are pretty low abundant. That means that conventional plasma approach would be blind for them. It would cut off somewhere here at 3 to 4 orders of magnitude of abundance, where the detector is just oversaturated by the high abundance signal. Now, one of the examples that I'd like to highlight here is BMP1. BMP1 is a very interesting protein because it actually plays a dual role in cancer. It can be tumor promoting, but it can also be tumor suppressive depending on its context. And what we saw here in our study as, again, like it's a pilot exemplification, but what we saw here is that we see some peptides being differentially higher abundant in the cancer samples. Here, for example, these bleeding peptide 1 and 2. And here you see the comparison between the cancer cases and the healthy controls. And some peptides that are lower abundant or higher abundant in the controls. And that may actually speak to the differential presence and function of different isoforms of BMP1 that may be indicative of how that protein works in the cancer. And that again is a good exemplification on how we can use the unbiased, untargeted proteomics to dive much deeper and go beyond the quantification of individual proteins, but tell about the function of the proteins and how basically certain proteoforms are changing and may affect your health state. So with that, I'd like to summarize by basically showing you this entire spectrum of the information landscape that we have in the proteome. So we start from the proteins, but proteins are not everything, right? Not everything that determines biological function. It's a proteoform. That means like what type of isoforms do we have? So in other words, what type of peptides that map to isoforms can we quantify? On top of that, and this is something that I have not shown here, but we have also some very interesting data showing that we can detect post-translational modifications with the unbiased mass spec readout in conjunction with particles. And of course, we are also able to see polymorphisms such as SNPs, which enables us to do proteogenomics based on the data. Then on the other end of the spectrum, you have protein-protein interactions, as well as the functional pathways to which proteins and other biomolecules map. And again, this is something where the granular information you gain on the PTM level, on the splice level, helps you to identify what pathways are really switched on and switched off. For example, phosphorylation is something that usually acts as a switch for protein activity. And we have published these results in 2020 in our Nature Communication paper. We also have one manuscript right now under revision, as well as other manuscripts that are currently prepared for publication. We have presented our platform at multiple conferences throughout the last years and engaged in collaborations around the globe with KOLs in proteomics, in clinical proteomics, and that are sharing our vision that there's so much more we can learn from plasma proteomics, and if we can do it at scale and depth. And with that, I'd like to thank you for your attention. I'm really looking forward to the discussion later and to the next talk. Thank you so much.
Thank you, Daniel. I will now invite you to leave the stage. Our next speaker is Robert Georgantas, Senior Vice President of Research and Translational Science at Biodesics. Robert has served as Senior Vice President of Research and Translational Science in Biodesics since August 2019. From 2014 to 2019, prior to joining Biodesics, Dr. Georgantas worked at AbbVie, where he served as Director of Immunology Programs and Biomarkers within the Genomics Research Center of Excellence, a group of experts tasked with applying genetics, genomics, epigenetics, and metagenomics to inform the product pipeline primarily regarding new target discovery, biomarkers for clinical trials, and asset positioning. Dr. Georgantas completed his PhD in pharmacology and molecular medicine at the Johns Hopkins University School of Medicine. But before handing over to Robert to begin his presentation, same as before, I'd like to share some poll questions. I'll show you the results so you can see how your response compares to the rest of the listeners joining us today. So what type of samples do you utilize the most in your proteomics research? Is it cell lines, tissues lysate, serum or plasma from large models, serum or plasma from small models, other biofluids, or others? You'll have around 30 seconds to vote. So moving on to the next poll. How many serum plasma samples does your group or would your group like to process and analyze per year? Is it more than 1,000 samples? Is it less than 1,000? Or are you just not sure? You'll have around 20 more seconds to vote. And finally, the last poll. What is the most important target for you when you plan for large-scale serum or plasma proteomic studies? Is it a large number of protein identifications? Is it analysis throughput? Is it study size, co-sample, or integration to other omics data? You'll have around 30 seconds to vote. I'll give around 10 more seconds. Perfect. So I would like to remind everyone that you can send your questions to Robert and Daniel using the question box on the panel on your screen. And now, without further ado, Robert, please join me on screen now by sharing your audio and video to begin with your presentation. Welcome, Robert. I can see your screen. We cannot hear you. If you can click that microphone button just on your panel. Can you try and click on that?
There it goes.
There you go.
Thanks,
Okay,
Naomi.
perfect.
Thanks for meeting me there, and thanks for the
No worries.
thanks for the introduction, as well as to Daniel for the great overview of the Seer technology. Today, I'll be taking everyone through the novel proteomic technologies that we're using at Biodesics for biologic translational science, as well as for biomarker discovery, with a particular focus on how we're integrating the Seer Proteograph technology into our current workflows and discovery platform. Now, I'll give a quick introduction to Biodesics. We're a data-driven diagnostic solutions company, and we split our business into essentially two arms. One being our commercial focus, which is focused on lung disease diagnostic testing. We currently have 7 diagnostic tests, 4 of which are focused on lung cancer diagnosis as well as treatment guidance, and then 3 tests that are focused on COVID diagnosis. We also have 3 pipeline tests, one that we call primary immune response that predicts outcome in lung cancer patients to treatment with immune checkpoint inhibitors. Another called risk of recurrence that predicts risk of recurrence after surgical removal of a lung malignancy. And then an NGS test, a rapid turnaround next generation sequencing test for therapeutic guidance. Now, while the commercial business is focused on lung disease, we have the other side of the business, our biopharma services arm, that is, essentially pan-cancer and pan-disease. We currently have 50 biopharma customers that we've, developed tests for in different cancers, including lung, bladder, renal cancer, and others, as well as in some autoimmune diseases. Now, while we're a technology agnostic company, we use genomics, radiomics, and proteomics to develop our tests. We're also somewhat proteomics technology agnostic as well. We're currently using a number of different technologies here, 5 different technologies, everything from high throughput ELISA assays to do targeted proteomics in a high throughput manner. Standard LCMS, also to do targeted proteomics. This offers high sensitivity as well as peptide protein ID. We have our proprietary deep MALDI platform that was developed over 10 years ago and has been improved over time to do more unbiased proteomics. So basically, instead of doing it a targeted panel, looking at a large section of the proteome. This is also a high throughput technique. And then we're incorporating 2 cutting edge technologies, the Bruker timsTOF platform for mass spec, which allows us to do both targeted or unbiased proteomics with high sensitivity in a very high throughput manner. We're also, and I'll talk about this extensively today, incorporating the Seer Proteograph into our workflows. So, again, doing unbiased proteomics in a high throughput manner. And then our strategy is actually to combine, the disruptive Seer Proteograph technology with the cutting edge Bruker timsTOF technology, to achieve, unbiased, high throughput, high sensitivity proteomics that allows us to do, protein and peptide ID that we can't do with our deep MALDI platform. Now, supporting those proteomics technologies, we have a few informatics technologies that we're using. One is our diagnostic Cortex AI platform. This is a platform that's been developed over the last decade and continuously improved specifically to answer clinical diagnostic questions. And within those supporting technologies, we've also developed a correlative protein set enrichment analysis technique that lets us look at, higher level pathway-based biology of what our tests are doing. And just recently, we've developed a computational technique that we call exact Shapley values. And what this allows us to do is it allows us to break the black box of the Diagnostic Cortex AI platform to explain at the patient level exactly how the AI platform is making a decision in the diagnostic test to make a call. Now, we've been a leader in blood-based clinical proteomics for more than a decade. We started out with our deep MALDI platform, with it through a combination of high shot MALDI with computational algorithms to do signal to noise ratio optimization as well as spectral alignment. In our first generation, we were able to look at about 100 proteins replicably. Even though we could see many more peaks in the MALDI spectra, we were able to look at about 100 proteins in their post-translational modifications, with CVs of less than 10%, which is the level that we believe we need to make a good diagnostic. With that technology, we have that technology was proved with our commercial product Veristrat, as well as a biopharma product that we developed with the Veo Oncology. Our second generation that we've been using for the last couple of years, moved to 274 proteins through changes in instrumentation as well as improvements in the computational algorithms. Our current pipeline products, risk of recurrence and primary immune response, are based on that second generation, along with some of our biopharma partner products that we've developed with Genentech, EMD Serono, Pfizer, and as well as other companies. Over the last 18 months, we've moved to our third generation of deep MALDI, which once again, based on improvements in instrumentation. Here we're using the Bruker RapidFlex, along with improvements in the computational algorithms that we're using for deep MALDI. We're now looking at about 1,500 proteins in their post-translational modifications in our third generation. But of course, even with this improvement and moving to our third generation, we're always looking to the next generation, to push over, more into the big data proteomics, keep pushing into large proteomics of 2,500 proteins and above. And the strategy that we're taking for the next generation is to combine the Bruker timsTOF platform with the Seer Proteograph technology. Now, what do we do with this data? So this data that we generate with these different techniques is used to feed highly reproducible data, robust, highly reproducible data into our Diagnostic Cortex AI platform. Now, like I was saying, the Diagnostic Cortex platform is, it's a AI platform that's been developed over the last decade and continuously improved, specifically to answer clinical diagnostic questions. We have components of the AI platform that do things very specific to avoid overfitting and make the test generalizable, so that rather than overfitting the model and having your data work with only the development set, we've tailored this platform to work with biologic data, so that not only does the test that the platform discovers fit the development set, but also validates in validation set after validation set after validation set. And once that data goes to the diagnostic platform, it's trained to find a diagnostic test that makes a meaningful clinical difference, looking at something like a sensitive versus resistant to a drug, or other phenotypes that we want to look at. Now, we have a number of technologies that we use. We commonly employ bottom-up proteomics or peptide analysis. We've established clinical diagnostic utility with standard LCMS here, with our Notify XL2 test, which is a test that helps physicians determine if a suspicious lung nodule is benign or not. The advantages here that this is a quantitative and precise technique. It can be multiplexed to look at small panels of proteins, and does away with the need for antibody development. Now, the disadvantages of this technique, while we can look across the entire proteome, that entire dynamic range that Daniel was talking about earlier, from things from low expression proteins to high expression proteins, It has the disadvantage that, using things like MRM and, SRM assays, that we can look at we can only look at small panels, relatively small panels, as well as, that it takes a significant amount of development time to develop those assays. We're going to talk today a lot about the unbiased approaches that we're using. One unbiased approach that we're using is our proprietary deep MALDI platform. And here, we can get, using deep MALDI, which is a very optimized, MALDI-TOF platform. We can get 5 or 6 orders of magnitude down in concentration over the plasma, proteome continuum. Here, represented by this blue box, the things that we assay with deep MALDI are the classical plasma proteins down into the tissue leakage proteins. So, here are things like serum amyloid A, the complement proteins, all the way down to things like the S100, proteins that are, biomarkers of, immune response. But what we'd really like to get to, and also, that platform has been verified by, the clinical utility has been verified with our and established with our VeriStat tests. But what we'd really like to see is an unbiased approach that gives us, a broad survey of the plasma proteome in a high throughput manner that's also highly reproducible. And that's really where the Seer technology comes in for us. Internally at Biodesics, we're now using the Seer technology in 2 manners. And the first way that we think about that is that the Seer technology allows us to increase the biologic resolution of our already established tests and tests that we're developing with deep MALDI. Now, deep MALDI is a fabulous technique in that it's highly reproducible, it's highly sensitive. I showed earlier that we're achieving now about 1,500 proteins, as well as post-translational modifications of those proteins. But the disadvantage of deep MALDI is that we don't have a lot of biological resolution. For those MALDI features that we're looking at, we're seeing, only a handful of those peaks do we actually know what protein we're assaying. We can use things like our protein set enrichment analysis to correlate. It's a correlative approach, looking at pathways that are correlated back to the deep MALDI spectra. So we can look at cancer-associated pathways like wound healing, different types of immune responses at the pathway level. We've also developed this technique at exact Shapley values so that we can break the AI black box and actually explain what our algorithm is doing at the patient level as well as at the global level. And then we think that through a combination of using the Bruker's timsTOF, it's high, it's very high speed mass spec and its exquisite sensitivity, along with the Seer Proteograph technology, that we can use those to get really fine biologic resolution and start understanding, the biology behind what deep MALDI and the diagnostic cortex is showing us by combining all of these. So what does that look like? So that looks, the workflow that we're using is that for our existing tests and tests that we're developing, we take the patient cohort, we use deep MALDI in the diagnostic cortex to segment those patients out into meaningful outcome groups. We can then use exact Shapley value analysis to go and break those subgroups down into feature groups to see if there are any differences in how the diagnostic cortex is going and making a call for a specific subject. This is an example from a study that we've done looking at one of the classifiers that we've developed, where we see at least 3 different ways that the diagnostic cortex is making a call for a bad outcome group. This is a group that's resistant to an immune checkpoint inhibitor. And our current working theory is that group 1 here, group 2 here, and group 3 here are actually showing us different molecular mechanisms that are leading to resistance to the drug. So we can further subset these outcome groups into potential biologic molecular mechanisms. And then use the Seer Proteograph technology along with the timsTOF platform to generate protein expression data. Protein expression data that is quantitative as well as we can do protein ID with these technologies. And that then allows us to do, standard data analysis. So, go and do differential protein expression analysis, do different types of analysis to subset those and tie those back to the outcome groups or the molecular groups. And then take that data and do bioinformatics, things like pathway analysis and upstream driver analysis to really dig deeply into the biology of what our tests are showing us are differences between these outcome groups. The other way that we're looking at using the Seer technology, the Proteograph technology, is for discovery of new diagnostic tests. So, very similar to what we do with our deep MALDI technology, take the patient cohort, generate protein data, protein expression data, feed that to the diagnostic cortex, and allow it to go and generate a test, a clinically relevant test based on, the Seer Proteograph data. And with that, we've been doing some background studies, some initial studies looking into, how amenable the Seer Proteograph technology is, for these types of studies. And the first question that we asked was, how deep can we get into the peptidome using, our current workflow, our current mass spec workflows, and as well as the other question that we're asking here is, how necessary are the different nanoparticles for, getting to a certain protein depth? And here we can see, in this experiment, this is looking at healthy volunteers using a DDA method, a data-dependent acquisition method, and a 30-minute gradient. We're seeing, a total number of peptides for each of the nanoparticles ranging from anywhere from 3,200 peptides all the way up to a little bit over 4,400 peptides. So, for a total, if we go across these, just under 10,000 peptides across all of the different nanoparticles combined, showing us that we can get to a significant depth within the peptidome. Now here, looking at this upset plot, which is, essentially a multi-dimensional Venn diagram, we can ignore all of this on the right-hand side. What we want to look at is we want to look at those with the single spot, because what that's showing us is that, in this single spot, for example, with nanoparticle number 5, what we're seeing here is that out of the 4,108 peptides detected from nanoparticle 5, 1,135 of those were uniquely identified only from that nanoparticle. And as we go down here in the number that were identified, nanoparticle 2 had 10,081 peptides, unique peptides identified, all the way down to nanoparticle 1, which had 635. Now, if we work out the numbers of those, we have 4,630 peptides, or 49% of the peptides that were only identified by a single nanoparticle. Showing us that we actually need to get to that protein depth, or that peptidome depth of just under 10,000 peptides, that we actually need all 5 of the nanoparticles contributing to get to that depth. The next question that we asked, and this is a little bit of a busy slide, was, how reproducible, is peptide isolation from the Proteograph technology? Now we did what we're looking at here is we're looking at, for each nanoparticle, the number of peptides identified, as well as the number of so-called public peptides identified. Now these are the peptides. The public peptides are those that are consistently, pulled down by that specific nanoparticle. So we did a number of replicates of these. And you can see here that for nanoparticle 1, the peptides in each replicate that were identified varied, but that across the board, we were consistently able to get 2,755 peptides on every single replicate. Now we did a lot of background work because, with data-dependent acquisition, it's known for having, a certain level of public peptides that the actual technique, the mass spec method can achieve versus those with something like the nanoparticle. So we did a lot of background work looking at, what is the absolute best, percentage of public peptide IDs that we could get with the technique, and that came up to 79.3%. So as these numbers here, approach 79.3%, they're approaching the maximum that we could, detect with the Seer technology. And what we find with this is that, if we go through and we take these percentages and, divide them by 79.3, what we're actually seeing is that we're seeing that, about 20% of the loss of, public peptides is due to the data dependent acquisition method, and that any additional loss from the nanoparticles is minimal. What we're actually seeing is that 95% of medium reproducibility of the public peptides pulled down by the nanoparticles. So, we're concluding from this that, each of the nanoparticles is highly reproducible in the proteins that it pulls down at the peptidome level. The next thing that we asked was how reproducible was protein detection. Here we're using very, very stringent conditions to look at the protein IDs, and what we're seeing is we use a number of healthy volunteer plasma donations. I'm showing here two representative pools, pool 1 and pool 3. And what we're seeing here is that we have, anywhere from between these different pools, from nanoparticle 4, around 300 protein IDs up to a maximum with, nanoparticle 5 of, just around 500 protein IDs. But the salient thing to look at here is really the reproducibility. We're seeing extremely, very, very low CVs of protein IDs with the different nanoparticles, ranging from, 1.8% to 5.9% with plasma pool 1, or 2.2% to 4.6% with, plasma pool 3, and are consistently seeing these types of results. So, we're concluding here that, the protein IDs that we're seeing and the number of proteins are, reproducible across the different nanoparticles. And then we can look across.
Hi, Robert.
What's that?
Oh, sorry to interrupt. I just want to say that we're running over time a bit, so if you can just move to the last slide for me.
Sure, absolutely. And I'll just say, these are the number of total proteins, that we were able to detect, as well as the CVs of those detections, running from, about 700 proteins to 800 proteins, depending on the different plasma donor with low CVs. And I will go ahead and go over this slide really quickly. So what we're concluding, in our preliminary studies is that, the Seer Proteograph technology, it gives us a broad proteome coverage. One thing that I didn't show, because it would have just been a gene list, is that when we actually look at the genes that we're interrogating, or the proteins that we're interrogating, we're seeing that broad spectrum of, that broad survey of proteins, everything from, cytokines and chemokines like CCL5 up to tissue leakage proteins. Specifically, we're seeing a lot of the S100 proteins, particularly S100A8 and 19, as well as the classical plasma proteins. We're seeing a deep proteome coverage. We're seeing reproducible data with low variability, making us think that this Seer technology is going to be, applicable for, our diagnostic Cortex AI platform and test discovery. And then the last thing I'll talk about is really, what are the applications for biopharma studies? So, just like, Biodesics is doing, it's applicable for, proteomic biomarker discovery, the underlying biology of a proteomic signature, as well as things like new drug target discovery, biomarkers of drug action and mechanism of action, stratification of patients by therapeutic response, subsetting of patients to look at disease basic biology, as well as studies into potential drug combination rationales. And I'll just conclude by saying that I've introduced Biodesics as a data-driven diagnostic development company. I hope I've shown how the Seer Proteograph technology is being integrated into our current workflows. Our early work with the Seer Proteograph is rather compelling. We think that it's going to be a stable, reproducible technology that we can use. And also that the Seer Proteograph technology would provide a great avenue for a broad range of biopharma studies. And with that, I'll pass back to Noemi, and so we can start the Q&A session.
Thank you, Robert. Feel free to stay on the stage with me while I introduce the next part of the webinar. And as you said, we will now be moving on to the Q&A session. So I just want to remind the audience that if you would like to submit a question, please do so using the question box. And if it's for a specific speaker, please indicate who your question is directed to. But before starting with the Q&A, though, I would like to launch the last 3 polls we have today. So I will now launch the first poll. So what stage of development or pipeline are you focused on? Is it research? Is it preclinical? Is it phase 1, phase 2, or phase 3? You will have around 15 seconds. And I'll be closing the poll in around 5 seconds. So moving on to the second poll. How likely are you to use proteomics in general for clinical research or biomarker discovery? You will have around 10 more seconds to vote. And finally, the last poll for today. How likely are you to use the Seer Proteograph technology for clinical research or biomarker discovery? You will have around 5 to 10 seconds more to vote. Thank you everyone for voting. So I'd like now to invite Daniel to join me and Robert on stage so we can begin with the Q&A. So Daniel, please join me now on stage by sharing your audio and video.
Hi.
Hi Daniel, how are you? So thank you for joining me both for the Q&A session. So I see we have some questions coming through. Daniel, I think this one's for you. Can you confidentially discriminate or quantify the mutant versus the wild type alleles of the same protein?
So conceptually, since the particles differentiate between proteoforms, or they bind at the proteoform level, you might have differential binding of mutant alleles, which can be one way to identify those. On top of that, so that would be a pattern you can derive directly from the particles. The mass spec readout, since it's not biased, it's untargeted, as long as you find the peptide that contains a mutation, you also directly read that out. So you have basically 2 ways. You can find mutations by differential binding on the particles, as well as like the mass spec sequences, like the proteins, right? The individual peptides. And if you find the peptides on your mass spec run, then you have a basically clear identification of those alleles.
Thank you. And Robert, I think this one's for you. Are there any publications on correlation protein set enrichment analysis and on Shapley values?
We do have 2 publications on protein set enrichment analysis, and the Shapley values are currently under review. We have 1 paper using Shapley values with Veristrat, and we've just submitted a paper of the actual Shapley value method. So if anyone wants to send an email to me at Biodesics, I'm happy to forward those references on.
Great. And have you used this technology to test preclinical species such as rat or monkey plasma proteome?
That's a question for me, I guess. Yes. So Yeah, we have internal work that shows that it works, and there's no reason to assume it wouldn't. Especially the plasma composition and the challenge statement of having too many of the high abundant proteins obscuring the signal is the same across species. The nice thing about the particles is that they do not require you to raise antibodies against like particular subsets of the proteome in order to deplete or like to do the job. So they are basically binding hundreds or thousands of proteins, and that's why they actually are by design easy to translate to other species, mouse or rat or whatever you can think of.
Thank you, Daniel. And I think there's another question for you here. So is the Seer proteograph primarily used for protein identification, or can it also be used for targeted protein quantification?
So that's an excellent question. And so it is, the Seer proteograph works very much upstream of whatever you do downstream with your samples. So the current product is designed with a mass spec readout in mind. So you end up with your peptides that you can inject into your mass spec, but the particles enrich and basically, or better to say, compress the dynamic range of all the proteins. And then conceptually, whatever you use as a readout technology, and it could be a targeted MRM approach, it could even be an ELISA, would be amenable to that platform, because yeah, you basically, whatever happens downstream of the proteins would benefit from the compressed dynamic range and detectability.
Thank you. We're running out of time, but I have a last question. So when we will be able to use the new Biodesics test using the Seer technology? And will this test be focused on lung cancer or other diseases?
So we're actually, if you contact our business development department, we're currently, taking, biopharma partners and offering the Seer technology to biopharma partners. Any diagnostic test, that we develop internally with the Seer technology would be, years down the road, from, discovery development, and then commercialization. That's usually a 2 to 3 year path, from discovery of a test. But, for, biopharma studies, we're currently offering the Seer technology to potential partners.
Okay, thank you. So as I said, we're running out of time, but all unaddressed questions from today will be passed on to Biodesics so they can reply to you directly. Thank you, Robert and Daniel, for joining me today. And thank you also to the audience for submitting those great questions. Please feel free to now leave the stage.
Great, thanks everyone.
Have a great day, bye-bye.
This is all the time we have, but I hope you enjoyed the session today. As I said, all unaddressed questions from today will be passed on to Biodesics. Also, please look out for an email tomorrow from us, as we'd love to hear some of your feedback on today's webinar. If you do have any follow-up questions or comments, please email this to the email below, and I will get back to you soon. And on behalf of Biodesics and CDX Engage, thank you all for your time, and I hope you enjoy the rest of your day. I hope to see you next time.